Hello, welcome back. We are continuing our journey through chapter 6. We are on to oxygen requirements. So we are going to reach back a lot to chapter 5 actually, as we're continuing with our chapter 6 material. So remember, for our aerobic cellular respiration, think about what the role of oxygen was. What was its job? All right, did you remember? Oxygen served as the final electron acceptor. It was the last step in the electron transport chain. So all the electrons that got stripped away from the glucose, the original molecule glucose, and put temporarily onto the NADH and FADH2, which were carried to the electron transport chain, which then got oxidized. They lost their electrons to the electron transport chain. Those electrons bounced down the electron transport chain, pumping protons, making the hydrogen ion gradient. And then oxygen, which is a very electronegative molecule, attracts those electrons, pulls in with some hydrogen, making water. That was the role of oxygen as the final electron acceptor. Now, when aerobic organisms do that, that's how they drive their ATP synthase. If you're an aerobic organism and you don't have oxygen, you don't have that final electron acceptor, your whole electron transport chain stops and basically the cell dies. And on a large scale, if a lot of your cells die, you die. Or if you're just a single-celled bacterium, you die if you need oxygen as a final electron acceptor. There are quite a few organisms out there that don't require oxygen as the final electron acceptor. And in fact, if they do grow in the presence of oxygen, they die because the oxygen to them is toxic. So we're going to take a look at a kind of a wide range of oxygen requirements in this little mini lesson. Okay, so you have these pictures here of tubes. This is a special type of media that it's semi-solid, which means it's not as hard as like a regular agar. It's a little bit more squishy, but the characteristic of the media is that closest to the top of your the surface, you're going to get high oxygen concentrations, and then it's going to diffuse into the media until way down towards the bottom you get very low, if any, oxygen. So it allows oxygen to diffuse through this media in a gradient. So the closest you are to the top is all of your oxygen, your highest concentrations, and the further away from the top, you have the anaerobic conditions. So the two easiest ones to identify are obligate aerobes, means they can only grow in the presence of oxygen. So we see our thickest growth up here at the top because that's where the O2 is. You get down further, there's not enough oxygen to serve that role as a final electron acceptor, and the organisms cannot survive. So obligate aerobes. In the next tube, we can see obligate anaerobes. There's no growth anywhere until it's way down at the very bottom where there's no oxygen, because any of the organisms up here, right, if they got inoculated, they just died because they were grown in the presence of oxygen. Let me tell you also, I forgot to mention this, how this media is inoculated. I'll erase all these little bacteria. What happens is you take what's called a needle. It's like a loop, but instead of a loop, it's a needle. So it's got a handle on it, and it's just got a thin wire. And so what they do is they get the bacteria on the needle, and they stab it straight down all the way to the bottom, and they pull it straight back up. And so you're putting bacteria into this tube straight down and straight up. So you get bacteria along the entire column of this agar. And wherever we see growth, that's where the bacteria were able to thrive, survive, and thrive. So even though, you know, we stabbed this organism all the way to the bottom, only the bacteria at the top grew. We stabbed this organism all the way to the bottom, only bacteria at the very bottom grew. For these guys, we stab it all the way to the bottom. This group is called facultative anaerobes. Now, looking at that distribution of the bacteria, where do we see more growth? Where's our highest concentration of organisms? Well, that's easy. It's near to the top. But we do see some growth down towards the bottom, but not as much. A facultative anaerobe has the ability to live in the presence of oxygen, and if there's oxygen, they can use oxygen as the final electron acceptor, and then they grow really well because they get 38 ATPs per glucose. Down towards the bottom, when there isn't any oxygen, they can still survive, there's no oxygen, so they can't do aerobic respiration, but maybe they can do a fermentation pathway where they get 2 ATPs per molecule of glucose. So here we get 38, and here we get 2. So clearly the cells that can make more ATP per molecule of glucose are going to reproduce faster and make more of themselves, whereas the cells down at the bottom are only, pumping out 2 ATPs per glucose. They're going to go a little bit slower. So that distribution makes sense based on their oxygen requirements. They can use oxygen if it's there, but if it's not there, they can still function, just slower. All right, this last group is called an aerotolerant anaerobe. Now, these guys truly are anaerobes. They cannot use oxygen, but they can live in the presence of oxygen. So again, we're going to stab straight down and straight up. So we're getting bacteria all along the length of that tube. Do we see any differentiation with the concentration of oxygen? No, it's evenly distributed. So oxygen is neither a help nor a hindrance for these aerotolerant anaerobes. They're truly anaerobic that can live in the presence of oxygen. We're going to talk about what they're going to have to allow them to do that. All right, this last group I don't have a slide tube for, so I'll draw one. These are called microaerophiles. I'm going to put microaerophiles over here in this area. So a microaerophile is an organism that can withstand oxygen, but it needs it just a little tiny bit, but too much is toxic. So they prefer like 2 to 10% oxygen levels. So they're going to grow probably maybe like right around here. So that's where you're going to get all of your bacteria. Too close to the top is going to be too toxic. Too far to the bottom is going to be not enough. So we would probably see some bacteria like a little strip close to the bottom, but not all the way at the bottom. So they're called microaerophiles. So these are the 5 categories of oxygen requirements, if you will, how these titles, these categories of bacteria based on their need for oxygen or the need to not be around oxygen. So you might be asking yourself, well, what is it about oxygen that some organisms need it, some organisms die in the presence of it, and some really don't care? Well, let's take a look. It has to do with enzymes. And what happens to oxygen in the process of cellular respiration. So remember oxygen, this final electron acceptor. So remember we had all of these proteins. Here's our plasma membrane, and we had our ATP synthase, and it spun around making ATP. Well, oxygen, when it's down here, this 1/2 of O2 that's eventually going to be water, if we have these electrons kind of bouncing down and eventually hook onto that oxygen to make water, sometimes those electrons are carrying extra energy. They didn't release all of their energy pumping protons, so they're a little bit more energized when they land onto that oxygen to try to make water. And so these are what we call toxic forms of oxygen. They're not good. They're called free radicals as a kind of a general name for them. And there's 4 listed here. So the first one is called Singlet Oxygen. And you don't need to know these molecular formulas, just the names would be sufficient enough. So in Singlet Oxygen, the molecular oxygen kind of has a higher energy state. Electrons have a higher energy state. These singlet oxygens are typically neutralized, is what we say, kind of taken care of by your normal antioxidants that are part of your cells, whether you're a bacteria or a human. Antioxidants that come in like fruits and vegetables, especially like orange veggies like carrots and squash and dark green leafy things, they have pigments or molecules called carotenoids, however you want to say it. These are naturally occurring antioxidants, and they have the ability to neutralize and reduce that energy state of these singlet oxygens. So it's not a huge problem for us. Our normal existing antioxidants work to take care of that. The next one is called a superoxide radical. So a superoxide radical is when in the process of this, electron transport chain, the normal, day-to-day actions of your mitochondria or you as the bacterial cell, you end up producing this hydroxyl radical, this O2-. So it's again got extra negative charges to it. And what happens is it produces no, so to get rid of it, aerobes have this enzyme called superoxide dismutase. So this O2 negative, so here's your superoxide radical right here, along with this enzyme and a couple hydrogen ions, the H pluses, they can convert that really nasty superoxide radical into hydrogen peroxide, which is less dangerous. We have ways to get rid of hydrogen peroxide pretty readily, okay? So superoxide radical is neutralized by the enzyme called superoxide dismutase. However, in that reaction of superoxide dismutase, sometimes those hydrogen peroxides become what's called the peroxide anion. That's this next one. Oops. My yellow check mark. There we go. Peroxide anion. So the peroxide anion is a byproduct of a incorrect superoxide dismutase reaction. So now we have this peroxide anion, and we have two enzymes that can neutralize it. We have the first one, which is called catalase, and the second one, which is called peroxidase. And in either one of those, these hydrogen peroxides are turned into pretty much water. The catalase reaction produces oxygen in addition to the water, or NAD+, which cells use anyways. Both of those are safe for the cell to have water. Obviously, oxygen gas is fine and NAD+ is fine. So catalase and peroxidase have the ability to neutralize the peroxide anion, which is a product of the superoxide dismutase reaction. This picture here in the slide, you'll be doing this in one of your lab activities, is you get a little culture, maybe a smear of your bacteria. You can put it on a slide or you can just put it right in the plate, and you actually drop hydrogen peroxide directly onto the bacteria. And if it bubbles, it lets you know, this is one of the biochemical tests that you'll be running, it lets you know that the organism contains catalase because it's taking that hydrogen peroxide that you drop on it, breaking it down into the water and the oxygen gas. So those bubbles that you're seeing are oxygen gas. Okay, and then the last radical or toxic form of oxygen is called the hydroxyl radical. And this is usually formed, let's see, I have to look this up. Hydroxyl radical is formed from ionizing radiation like gamma rays and X-rays and things like that, and an incomplete reduction of the hydrogen peroxide. So again, an inadequate peroxide anion reduction. But because catalase and peroxidase work really well, we don't typically see the hydroxyl radical very often, and we don't have a lot of exposure to ionizing radiation, luckily. And so this one, we just don't see too much. So we're going to focus in this class primarily on the two types of toxic forms of oxygen that we have enzymes for, so superoxide dismutase, catalase, and peroxidase. So when you think back to the last slide with all of the different tubes, obligate aerobes, for them to be able to live in the presence of oxygen and let oxygen be their final electron acceptor, they have to have these enzymes be part of their normal cellular processes, because without them, they would get ruined by these toxic forms. So obligate aerobes, facultative anaerobes, and probably some of those aerotolerant anaerobes, they are going to have some combination, if not all, but maybe a couple of these enzymes to help neutralize those toxic forms of oxygen. Obligate anaerobes probably lack any of these enzymes. So even though they might have been stabbed into that media, on the top where there's oxygen, oxygen is a great final electron acceptor. And so it's probably doing its job there at the top of that obligate. So if we had our tube, right? Here's our inoculation. So there's bacteria, say, trying to grow. But if these are the obligate anaerobes, up here, all these bacteria that are using oxygen as a final electron acceptor because it does a really good job, they're building up all of these toxic forms. They're going to be shooting off superoxide radicals and peroxide anions and hydroxyl radicals, and there's no way to fix those. And so all of those cells living towards the top of that tube die. And the only ones that can survive are the ones that are down here because there's not any oxygen. They use something else as their final electron acceptor to allow them to complete their metabolic processes. All right, I think we are done. Okay, so that is a little mini lesson on oxygen requirements. We're going to move on to other, like nitrogen and other growth factors and stuff. All right, see you next time. Bye.